Hello friends, welcome to this session. So in this session we are going to talk about one of the important biomolecules of the living system, carbohydrate. In that especially this TCA cycle, it's a central pathway of metabolism. Course code is 23U FSC 404 and the course name is Allied Biochemistry 2. Course objectives gain knowledge on different sources of biomolecules, learn the metabolism and biosynthesis of essential nutrients, then understand the different deficiency disorders. Course outcome recall the biochemical functions of various biomolecules and hormones, illustrate the general metabolic pathways of macromolecules. So carbohydrates, they are organic compounds made up of carbon, hydrogen, and oxygen. 1:2:1 ratio. General formula CH2O. Primary source of energy, structural role, and example glucose, which is there in human metabolism. Monosaccharides are classified into based on their number of carbon atoms: trioses, glyceraldehyde; pentoses, ribose; hexoses, glucose and fructose. By functional group, aldoses, example glucose; ketoses, example fructose. This is fructose. Then monosaccharides, they are having open chain in cyclic structure. Here open chain is converted to cyclic structure here. Alpha, beta anomers are possible. Glucose is an aldohexose. Fructose is a ketohexose. Real life biomolecule, that is 5 carbon sugar, is ribose, which is present in DNA and RNA. This is the linear structure of fructose and the ring structure of the fructose. The carbohydrate metabolism is a very fundamental biochemical process that ensures a constant supply of energy to the living cell. This is the central metabolic pathway associated with the formation and breakdown of carbohydrates into energy. The most important carbohydrate which is used in our metabolism is glucose, which is broken down into pyruvate via glycolysis, then enter into TCA cycle or Krebs cycle. That is always coupled with oxidative phosphorylation in order to generate ATP molecule. In carbohydrate metabolism, we have several pathways: glycolysis, citric acid cycle, gluconeogenesis, glycogenesis, glycogenolysis, hexose monophosphates, and galactose metabolism. Now we are going to see about citric acid cycle or tricarboxylic acid cycle or Krebs cycle. This is overview where you can see that other pathways are there, but we are concentrating on here TCA cycle, where pyruvate converts to acetyl-CoA. Acetyl-CoA enters into TCA cycle, it is coupled with oxidative phosphorylation. The fate of the pyruvate, I see that pyruvate is converted into ethanol in alcohol fermentation, or it is converted into lactate, or it may be converted into carbon dioxide water when enters into TCA cycle, citric acid cycle. Citric acid cycle is a very, very important one. The end product of glycolysis is pyruvate that is transported into mitochondria and loses the carbon dioxide to form acetyl-CoA. Krebs cycle occurs inside the mitochondria and generates a pool of chemical energy. When acetyl-CoA is oxidized to carbon dioxide, that is CO2, in the Krebs cycle. So what happens is that in the entire chemical energy which is released in the form of three molecules, that is the one is three molecules of NADH, one molecule of FADH2 and one ATP molecule. So the cycle starts and ends with the only one molecule that's called oxaloacetate. This cycle is also called as Krebs cycle because Dr. Krebs explained it, and citric acid cycle or tricarboxylic acid cycle, TCA cycle. This cycle utilizes about two-thirds of total oxygen consumed by the body. TCA cycle is strictly aerobic in contrast to glycolytic pathway. Oxidation of 3-NADH by electron transport chain coupled with oxidative phosphorylation results in 9 ATP. FADH2 provides 2 ATP. Total of 12 ATP molecules are produced from one acetyl-CoA. And this is the entire pathway, the TCS cycle. You can see that we'll start from here. Acetyl-CoA which comes from pyruvate. Acetyl-CoA combines with oxaloacetate and citrate synthase enzyme, which is converted into citrate. Citrate again converted into isocitrate by the enzyme aconitase. Then isocitrate is converted into alpha ketoglutarate by the enzyme isocitrate dehydrogenase. Then alpha ketoglutarate is converted to succinyl-CoA where NAD is converted to NADH plus H plus and CO2, and you see that you are getting succinyl-CoA. Succinyl-CoA is converted into succinate by removing one molecule of GTP and coenzyme A, and you are getting succinate here. Succinate is converted into fumarate by conversion of FAD to FADH2, and you are getting 4-carbon molecule called fumarate. By adding water, you see that by the enzyme fumarase, malate is formed. Malate is converted to oxaloacetate by converting NAD to NADH plus. Malate dehydrogenase is the enzyme, and you are getting oxaloacetate again. Oxaloacetate once again combines to another molecule of acetyl-CoA. This cycle repeats. This is how it is repeated several times. That is why it is called a tricarboxylic acid cycle. TCA cycle. And TCA cycle is always connected with or coupled with oxidative phosphorylation. It is the process in which ATP molecule is actually formed or generated as a result of the transfer of electrons from NADH or FADH2 to the molecular oxygen, that is O2, by series of electron carrier molecules called electron transport chain carrier molecules. They are present here, electron transport chain molecules in the plasma membrane, this mitochondrial membrane. It takes place in mitochondria, which is the major source of ATP production in aerobic organisms. Here, citric acid cycle happens here in the matrix of the mitochondria, and you can see that it is always coupled with electron transport chain or oxidative phosphorylation, where you are having complex 1, complex 3, complex 4, and the ATP synthase enzyme is there, where ADP inorganic combines and proton is pumped inside, ATP is formed. And see that we are having 1, 2, 3 complexes present in the plasma membrane, especially the inner membrane of the mitochondria. So that is why citric acid cycle is always connected or always coupled with oxidative phosphorylation. So during oxidative phosphorylation, you will get ATP molecules, because where you see that ATP is synthesized by this ATP synthase enzyme, which is present in the inner membrane of the mitochondria. And final acceptor of the electron transport chain is molecular oxygen. So the total ATP production from one molecule of glucose is here. Glycolysis net yield 1 ATP, 2 NADH plus gives 2 FADH2, 2 electron transport. So 2 ATP, 3 ATP. Conversion of pyruvate acetyl-CoA second stage, 2 NADH plus H plus is used, 5 ATP is formed. Stage 3 is TCA cycle. ATP at one site gives 2 ATP, NADH plus H plus at 3 steps. 2 electron gives 15 ATP. FADH2 at one step gives 3 ATP. So total ATP molecule, one molecule of glucose is 30 ATP molecules. So friends, let us come to this question and answer session. First one, which enzyme catalyzes the conversion of citrate to isocitrate in the TCA cycle? A, succinate dehydrogenase, B, aconitase, C, citrate synthase, D, malate dehydrogenase. The answer is B, aconitase. The next one is, in the TCA cycle, succinate is converted into fumarate by the enzyme A, fumarase, B, succinyl-CoA synthetase, C, succinate dehydrogenase, D, isocitrate dehydrogenase. The answer is C, succinate dehydrogenase. Next question, which molecule enters the TCA cycle by combining with oxaloacetate to form citrate? A, pyruvate, B, acetyl-CoA, C, succinyl-CoA, D, alpha ketobutyrate. The answer is B, acetyl-CoA. Next one, how many molecules of NADH are produced if from one turn of the TCA cycle? A, 1, B, 2, C, 3, D, 4. The answer is See, three molecules at one turn of the TCA cycle. And references as usual, principles of biochemistry by Leninger, biochemistry by Schreier, lipid coat illustrated reviews biochemistry, NCBI science director also there, and that is also giving you a lot of references. So with that, and now we have come to the end of this particular session. So thank you so much for your patient listening. Once again, and thank you. Let us meet again in the next video session. Thank you all.